Welcome to the Step Point of View Study Guide, and in this lesson, we learn about the pentose phosphate pathway and glucose-6-Phosphate dehydrogenase deficiency. The pentose phosphate pathway is also called the HMP shunt. It is used to synthesize NADPH used in reductive reactions like glutathione reduction in red blood cells and fatty acid and cholesterol synthesis. The HMP shunt also makes ribose used in nucleotide synthesis. ATP is not used or produced in this pathway at all. The pentose phosphate pathway occurs regularly in lactating mammary glands, liver, adrenal cortex, and the red blood cells. It occurs in two parts. The first is the irreversible oxidative part. Starting with glucose-6-Phosphate, glucose-6-Phosphate dehydrogenase reduces NADP+ and creates NADPH and 6-phosphogluconate. It is used to reduce yet another NADP+ in creation of NADPH and getting decarboxylated into ribose 5-phosphate. The second part is the reversible non-oxidative part. Ribose 5-phosphate is isomerized by phosphopentose isomerase into ribose 5-phosphate. Ribose 5-phosphate is what's used to build nucleotides. If nucleotide synthesis is not needed but energy is, then it can be converted into either fructose 6-phosphate or glyceraldehyde 3-phosphate by transketolase requiring B1 to enter into glycolysis. Glutathione is an antioxidant used by red blood cells to cleanse it of free radicals. It is synthesized indirectly by the oxidation of glucose-6-phosphate to 6-phosphogluconolactone by glucose-6-phosphate dehydrogenase. Then, glutathione reductase uses the NADPH generated to synthesize glutathione. Therefore, a deficiency of glucose-6-dehydrogenase results in a deficiency of glutathione, making the red blood cells not able to protect themselves from free radicals. This leads to hemolytic anemia, a condition in which the red blood cells are being destroyed faster than they can be produced. Fava beans, sulfonamides, nitrofurantoin, primaquine, chloroquine, anti-tuberculosis drugs, and even just having an infection can all increase free radicals in the body. Patients with a deficiency of glucose-6-phosphate dehydrogenase are thus more prone to hemolytic anemia from any one of these. Glucose-6-phosphate dehydrogenase deficiency is most common in African American populations because having this increases the resistance to malaria, similar to sickle cell anemia. It is caused by an X-linked recessive disorder. Heinz bodies occur due to excessive oxidative stress on the red blood cells. Bite cells result from macrophages in the spleen removing the Heinz bodies. That's all for this lesson, Step Point of View. We'll see you in the next video.